Thank you very much, Joachim. So now I would like to take you back to the basic material, so the plant materials that we have investigated in the GRACE project. And I will actually tell you the results on both the maize materials that have also been used in the animal feeding trials, but also on the potato materials that were initially planned to be included into the GRACE project as well. But I would like to start with acknowledging the contributions of the people who have been involved in this research. These are the people from RIKILT Wageningen UR, but also from Wageningen UR Biometris, from Wageningen UR Plant Breeding Group, and also from the University of Nijmegen, the Analytical Chemistry Department. Then I would like to first step back to the basic approach for the safety assessment of GMOs. Which is a comparative safety assessment, and this means that in each case we compare a GM variety to a non-GM conventional counterpart. And then you can basically split up the whole safety assessment into two parts. So the first part is aimed at assessing the intended effects, which is, for instance, the herbicide resistance or the insect resistance. And then in each case we also always assess the potential present unintended effects. And these unintended effects have been defined by EFSA as the differences between the GM plant and its appropriate control lines, which go beyond the primary expected effects of the introduced target genes. So how do we assess these unintended effects in practice? Well, up to 2013, this was basically done in this way. So we had 4 ways of looking at these unintended effects. The first approach is to look at the molecular characterization of the introduced genetic construct, but also at the flanking regions of the construct. Secondly, a detailed phenotypic analysis is performed of the GM line, but also of the conventional counterparts. Similarly, economic performance is done of the GM line and the counterparts. And finally, but very importantly, a compositional analysis based on targeted analysis that targeted all the key nutrients, but also the key anti-nutrients, natural toxins, allergens, etc. So quite a detailed targeted analysis already. And then in 2013, it was also a standard. It became a standard tool to perform animal feeding trials with whole foods to assess also for unintended side effects of a genetic modification. Now we know that this approach of the animal feeding trials is not very sensitive, and therefore it was already advocated before 2013 to develop more detailed, to develop methods to look more detailed into the compositional analysis of the GM varieties and the control varieties, and to do this in an untargeted way, so to do a very broad assessment of the physiological parameters of the GM variety and the control variety using omics technologies. And this has been applied within the GRACE project. So we have looked at transcriptomics, proteomics and metabolomics. So transcriptomics actually looks at the DNA being transcribed into RNA. So we look at the total RNA population of plant materials. In the case of proteomics, we look at the set of proteins in the plant material, and metabolomics is looking at the secondary metabolites in the plant. Now, the advantage of omics analysis is that in one analysis you can look at very many endpoints, and in this way you will get a very large data set on your plant material. Of course, at the same time, the disadvantage of omics analysis is that you get very large data sets. So then the question was, how can we actually analyze the data for meaningful differences, given the fact that, of course, there is a lot of natural variation between plants grown in the field, and you have to take this into account when you're actually looking for meaningful differences. And these differences can, this natural variation can relate, of course, first of all to the genotype, so if you have different plant lines, but also to the conditions of growth. For instance, you can have differences in the soil conditions or in climatological, so the weather conditions. So to solve this issue, we have been discussing a lot with the people from Wageningen who are biometrists and from the University of Nijmegen, the Department of Chemometrics. And on the basis of these discussions, we have developed a one-class model to distinguish, to identify differences between GM varieties and non-GM counterparts. And the basic criterion for this model was actually that if you have a profile of a variety that you cannot consider as safe, it should fall outside of this one class. So very schematically, this is what it looks like. In this picture, each dot stands for one profile, one omics profile from a variety that we consider as safe. And this may actually also include the parent variety in the case of a GM variety, because then the parent variety usually is also a variety that is commercially available and considered as safe. And then the next step is to assess the GM variety. And basically there are two options in that case. So the first option is that the GM varieties falls within the one class of varieties that we consider as safe. And the alternative is, of course, that the GM variety does not fit into this one class. And in this case, it doesn't mean that the GM variety is not safe, but it does mean that further assessment is required and additional studies may be needed to conclude on the safety of the GM variety. So this model had already been initially developed and has been published in 2013. But in the GRACE project, we have continued to work on the model, and we have further developed it and applied it at a much larger scale. Now, I can only be very brief about the model itself, but it's a SIMCA model, which stands for Soft Independent Modeling of Class Analogies. And this is a schematic overview of how it is developed on the left side and how it is applied on the right side. So it's developed on the basis of a training set. These are varieties that we consider as safe, that are commercially available. And this will also, in general, include the parent samples. And then in the next step, we perform a cross-validation step, also with varieties that we consider as safe. And on the basis of this cross-validation, we actually determine the outside of the model, and that is called the critical class distance value. And basically, if we have done this cross-validation, then we have our basic model, and then we can apply it. And in each case, we apply it first to test samples, which we also consider as safe, and which therefore are likely to fall within the one class that we have formulated. And based on these results, we can apply, for instance, experimental samples that can fall out either inside the one class or outside of the one class. And of course, also GM samples can be assessed and may fall either inside the one class or indeed outside of the one class. So this we have done for quite large numbers of data from metabolomics. So maize metabolomics data were generated by RIKILT, but also by CSIR in South Africa. Transcriptomics data were generated by RIKILT as well as by CRAC in Spain. For potato, we had metabolomics data that were generated by RIKILT as well as by CSIR in South Africa. Here I should say that we have some difficulties in analyzing these data, so we are still working on this one, but they will in the end be fully analyzed as well. And we have transcriptomics data from RIKILT. What I also would like to underline is that basically all these omics data, they can also become available to any of you who would be interested, and they can always be reanalyzed at a later time point. If you have some additional interest to look at the data, they are available and they can be reanalyzed and used by other groups. So this is one example of how the model is constructed and how it works in practice. So in this case, we had potato metabolomics data, and here we had 10 varieties to make the model. So the one is the parent variety, which is a commercially available variety, and we had 9 additional commercially available varieties to build the model. And then we had, for classification, we had 7 varieties, 1 GM variety, and also 6 additional experimental varieties. And here I should indicate that these experimental varieties were considered fit for human consumption, but they are not on the market yet. Here, the full data matrix was actually 44 profiles, with each over 100,000 variables. And these 44 profiles include these biological repeats that are in the last column. And when applying the model, we actually did two types of assessment. So it goes too far to go in detail, but at least this is the basic assessment to see whether one profile falls inside or outside of the model, and this is a more stringent approach. And here you can see that if you have the more stringent approach, then all of the experimental samples, which are fit for human consumption again, they all fall outside of this one-class model, which shows that the model is quite conservative, as it was intended to be. And at the same time, you see that the GM lines, the Phytophthora-resistant lines in this case, they all fall within the one-class model, and actually in both approaches. Here you also see that if you take a slightly less stringent approach, then also one of the experimental lines falls into the one class. So we did this for all the data. So we had, as I've just shown you, 44 profiles for potato metabolomics from RIKILT and 104 transcriptomics potato profiles as generated by RIKILT. For maize transcriptomics, we had data from RIKILT as well as CRAG. So from RIKILT, we had 16 profiles, 8 from CRAG. For CSIR, we had 36 profiles for maize metabolomics and 46 maize metabolomics data from RIKILT that have all been assessed in this way. And with that, I come to the conclusions from the GRACE projects on the plant omics. So based on the conclusions, we can at least state that unintended effect can probably be more effectively traced by using this type of omics analysis compared to the animal feeding studies with whole foods. And we base this on the following. So first of all, GRACE data can actually have shown that actually you can apply the comparative safety assessment as it is now being performed using the targeted analysis in exactly the same way based on omics analysis. And there you can, for instance, use the one-class model. And this means that in practice you can make the direct comparison between the GM variety and the conventional control plant, but in the same way you can also make the comparison to a whole range of conventional varieties. Now we have also tested some mycotoxin contaminated maize samples, and here it was shown that they are classified in all cases outside of the one safe class. I should stress here that these samples were separately sampled in Spain, so they are not related to the samples that, or to the maize materials that have been fed to the test animals. But this also showed that in such a case, this would be a basis for further analysis of this type of samples, because they are classified outside of the one class. Then all the maize varieties, whether GM or not GM, that have been fed to the test animals and that have been tested in the GRACE project were all classified as inside of the one class model. And indeed, the maize harvest for study C was not included. And also, I should indicate that in one case, we could not make a good enough model because the number of conventional varieties included in the system was insufficient. So there we would have to extend the number of varieties and redo it again. But where we had a good model, they were all indeed classified as within the one safe model. Well, I've just shown you that also for experimental potato varieties that are indeed fit for human consumption, but that are genetically more distant, they are also classified in most cases as outside of the one safe class. And this, indeed underlies the conservative approach. Then what we also did is we looked at whether these omics data can also identify foreign elements, so elements that are not known in the particular species. So we did this in a non-targeted approach, which means that we actually acted as if we did not know what was in there. And in this way, we could easily identify the foreign element in the maize, which is the CRY1AB gene. So also for newly expressed products, this approach is informative. So based on these observations, we can conclude that omics data as generated by the GRACE project does provide details of the plant material which can actually facilitate a non-targeted safety evaluation. And in this way, it actually provides a better basis for the decision on the scientific rationale of the next steps in a risk assessment compared to the performance of an animal feeding trial with the plant-derived whole food or feed. And of course, you cannot exclude that at some point it may be advocated to perform an animal feeding trial, but then you can do it on the basis of a clear scientific hypothesis. And with this, I would like to conclude my presentation and thank you for your attention.